There was not enough heat. And I think this time there might be much more heat. And I anticipate this type of situations on the floor, but maybe also within the audience. I don't know. Well, the structure is as follows. First, we have the talk of Patrick Couvreur, 20 minutes, followed by so that's the talk about the big progress of the field. The nano-progress of the field is after Patrick is Kinam, 20 minutes. Then we vote again. And then I have the slides with the statements. 5 statements, 10 minutes per statement. I will explain a little later how this will work. 2 statements are from Patrick Couvreur, 2 statements are from Kinam Park. And myself, of course I'm a moderator, I have no opinion, not at all. But I made a sort of surprise statement for the last part of the discussion. So I think I invite Patrick to come over. No, let's first vote. Please vote now if you have installed the link. Big progress or nano progress? Wow. Getting more complicated.
But it's a cool that you draw directly to the link. That it's easier.
Oh, it's easier? So,
yeah.
So they know what to do?
Yeah, it's with the phone.
If I have 1 minute more, don't.
No, I don't cut you. Let's wait until we have voted. The vote will come there? I don't know. Will the vote come there? Later, 10 seconds.
Okay.
10 seconds more for voting, and then we will have the outcome, if I'm correct. So the voting time is over. Let's see what is the outcome. It's coming now on the screen. I hear people need more time. I hardly see you. Do you need more time? Say yes. Okay, I will be patient. Very difficult for me.
Sácame de pantalla.
So now it's 10 seconds more because I need to close and to move on. If the figures don't move, I think then the voting is closed. But I see that it's quite a difference actually. And the figures are quite stable now. 10 seconds are over. Please stop and put away your mobile and focus on the talk of Patrick Couvreur. Please.
So I see that I have to be really convincing to change your vote. It will be probably not so easy. So I am very happy to share with you my view on this debate, big or nano-progress concerning the nanomedicine. First of all, I would like to be clear about what is the definition of nanomedicine. Nanomedicines are, in fact, medicines which are formulated as nanoparticles, but taken in the broadest sense. This means including, of course, liposomes, polymer nanoparticles, polymer micelles, iron oxide nanoparticles, metal oxide frameworks nanoparticles, etc., etc. But the goal is to improve the bioavailability, the pharmacokinetic, the biodistribution, and or the metabolization. And of course, it is for enhancing therapeutic efficacy and reducing toxicity of the encapsulated drug, eventually allowing imaging and diagnosis. I would like first to share with you the view of Roy Amara, who was saying for new or emerging technologies that we tend, as you can see here, to overestimate the effects of a technology in the short run. But we have also a tendency to underestimate, as you can see here, the pointer is not going more, sorry, to underestimate the effects in the long run. I don't know if you see the arrow on the screen. No, it's not working. And this is also the view of Gardner with a well-known, Gardner hype cycle for maturating a technology. You know that when you have an innovation, you have immediately a growing towards a peak of probably inflated expectation. But then after you have a decrease through a certain disappointment, but then you have a slope of improvement towards a maturation and a plateau of productivity. And I don't know where we are actually with the nanomedicine, and I will try to answer to this question today. And I would like to answer to some arguments which are generally encouraging the skepticism about nanomedicine. First of all, sentence like, translation of nanomedicine from bench to clinic is poor. Or there are very, very few nanomedicine FDA approved and on the market. And I completely disagree with that. Why? I was just looking here, as you can see, a table which was published by Mitragotree in 2016, with a lot, as you can see here, of medicine, nanomedicine FDA approved. And at the same moment in 2016, in another publication in pharmaceutical research, there was called that 51 FDA-approved nanomedicine existed and already 77 in clinical trials. And I would like to add that if you are looking to the number of nanomedicine in clinical trials, again, the table of Mitragotree, you can see that there are a lot. But now we are a couple of years after, and already 2 are FDA approved, including the Viexos and also the Patisiran, which are approved since that time. So this is very, in my opinion, encouraging. And also here, if you are looking to the evolution over the time of the number of nanomedicine in clinical trials, you can see that there is a very clear increase, as you can see on this cartoon. And no, I am not an economist, as you know, but if you are looking to some economical review, you can see here that actually there is almost a little bit more than 200 billion euro, which corresponds to the global market of nanomedicine, and more interestingly, as you can see here, the market growth rate is much higher for the nanodrugs, for the nanomedicines, than for the normal medicines. So you can see that there are marketed nanomedicine and a lot in clinical trials. So the second argument is the concentration in the tumor remains very low. Again, I disagree with that. Why? I would like to make a comparison with the antibody-based therapy. It was, you know, a very long way since the discovery in 1975 by Kohler and Milstein of the monoclonal antibodies. And we have to accept that antibodies actually in tumor therapy, this is a success. Although they do not distribute and accumulate into the tumors more efficiently than the nanoparticles. And of course, depending on the publication, the clinical trials, etc., in between 0.5% and 7% of the injected dose per gram of tumor are generally located, the drug is located into the tumor after nanomedicine administration. Of course, I agree that you can consider that it is low, but in relative terms, it is higher than the conventional anti-cancer compounds. And the last point, you know, the percentage of tumor accumulation is not always a goal in itself. And it is sometimes, but sometimes also not predictive of the anti-cancer efficacy. And I would like just to illustrate that by a very nice paper published by Dario Neri, at the ETH of Zurich. And he was comparing a small molecule drug conjugate, as you can see here, with an antibody drug conjugate. And if you are looking here, the concentration into the tumor, it is the small molecule drug conjugate which win. But if you are looking now to the efficacy, the preclinical efficacy in a mouse model, you can see that the antibody drug conjugate is much more efficient in blue than the small molecule drug conjugate in red. And I would like to continue this comparison with the therapeutic antibodies. Because with the therapeutic antibodies, you know, the patients which are treated are selected. And in fact, this is not the case with the nanomedicine. Of course, it depends on the antibody, it depends on the type of cancer you are treating. But without preselection with the antibody, the percentage of response is generally very low. In general, much less than 10%. But now, if you are able to select the patients on biomarkers, etc., then over 50% of the patients can respond to the antibody therapy. And this is a personalized medicine, as you know. And I think that we, and a lot of colleagues agree with me, that we have probably in the future to follow a similar approach to identify those patients displaying an APR effect and those not displaying an APR effect. And this is, again, the personalized medicine. If you have a cancer or a patient with an APR effect, then you can administer the nanomedicine. Otherwise, you have to change or to stop the treatment. And I would like just to give you one very clear illustration of that. This is in clinical cancer, a therapeutic essay which was done in 2017. And you can see the idea was to use iron oxide nanoparticles and to look the ability of those iron oxide nanoparticles to enter into the tumor lesion before to treat the patients with a liposome loaded with irinotecan. And in fact, depending on the concentration of the iron oxide nanoparticles in the tumor, the patients were divided into two groups. As you can see here, a group above the median, and also, as you can see here, a group below the median. And now, if you are looking the best change in tumor size, you can see that it was the case. We had a best change with a group of patients which was above the median in terms of concentration of the iron oxide nanoparticles, either 1 hour or 24 hours after the injection. Another argument encouraging skepticism is we are far from the dream of Paul Ehrlich. Well, I can agree with that. We have to be humble, and possibly, that too much hope was indeed based on the up-here effect and in general in the pegylated nanomedicine. But you know, this is true also with any drug, because you know that the attrition rate with any cancer compounds is also very high. And we can consider that over 10,000 drug candidates in preclinical trials, only one is able to reach a market. And in my opinion, the nanomedicine is not doing worse than the normal anti-cancer compounds. But probably look now to the future. We have to look for future directions, original directions, which probably don't take always advantage of the EPR effects. And one way is probably to use the local administration of eventually stimuli-responsive nanocarriers. And I have just picked two recent publications, one which was published in Science. It is a fantastic publication with the transcranial administration of lanthanide doped nanoparticles which absorb near-infrared light, able to emit visible light, and these allow neuron stimulation. And there was another example published in Nature Biotech about those discoidal micrometer nanoporous silicon particles able to release doxorubicin coupled with a polymer, which was able to, by reptation, by diffusion, to go deep into the tumor. You will tell me, but it is preclinical. Okay, then I will give you an example, a clinical example. And this is a possibility to use stimuli-responsive nanoparticles as radiation enhancers, especially the hafnium oxide nanoparticles, which are injected intratumorally, again, local administration, before radiotherapy. And there was a very interesting clinical trial done in 2017 with very, very interesting results, with a decrease of the tumor size for those patients who were treated before the radiotherapy with hafnium nanoparticles. But very recently, last month, in June 2019, at the Congress of the American Society for Clinical Oncology, there were disclosed very impressive data on upper aerodigestive tract cancer, and 9 over 13 patients had a total remission with a total disappearance of all the tumors. Other future directions which don't use the pegylation approach is to use endogenous carriers for the delivery of nanomedicine. And I would like just to give you one example coming from my group, and I spoke about that yesterday, where we used, in fact, LDL as indirect carrier by developing those squalane-based nanoparticles. And because the squalane moiety has a very strong affinity for the LDL, after administration, the nanoparticles disaggregate. The bioconjugate, the drug linked with the squalane, will insert it to the LDL, and this will explain why those nanoparticles, those bioconjugates can be, as you can see here, long circulating, and also target tumor cells hyperexpressing the LDL receptor. But I have another example of that in the group of Moa and Mogimi, who used erythrocytes as indirect carrier. And you can see here very clearly that this group developed nanoparticles able to stick to erythrocytes and therefore to prevent the macrophage recognition and the adverse cardiopulmonary distress. But probably that the more interesting example of that is the fact to use infiltrating T lymphocytes, or even now CAR T cells, that can be used to target nanoparticles, of course loaded with a drug, to disseminate the tumors. And you have an example here with autologous T lymphocytes, which are used to invade with their nanoparticle load, to invade, as you can see here in red dots, disseminated lymphoma. And there are also still other exciting perspectives, especially to use nanoparticles for blocking the immune checkpoints. And there was a lot of proposal in this way in the last session of the European Research Council applicants. To use nanoparticles, to interact with PD-L1 or with PD-1 and to hinder the tumor cell to escape from the recognition by the T lymphocytes. And I am sure, but this is not my good presentation, but this is sure that, Kinam, you will agree with me on that point, because at the opening ceremony, you gave the Journal of Control Release awards to the use of nanoparticles to target, T cells. So I think this is really a very interesting new approach that we could consider. But I would like really to come back also to the use of nanotechnologies for nucleic acid delivery. And this is a great news that the patisiran has obtained the FDA approval. This is a first RNA-based therapy for TTR amyloidosis. And this is thanks to the use of lipid nanoparticles. Otherwise, the RNA wouldn't work so efficiently. And this has changed the life of thousands and thousands of patients. And now this opened the door to a lot of ongoing clinical trial. At least I counted with 8 different messenger RNA. And you have also all the fields of the gene editing, where you can use, as you know, Cas9 fused with cytidine deaminase, which is able, of course, to induce the cytidine deamination at the level of the DNA and to do some mismatch repair. But to do that, of course, you have to formulate those very sensitive fused proteins to allow the protection from the degradation, from the metabolization, but also, of course, eventually the targeting to the good cells. But until now, we spoke about cancer, because that's true that most of the nanoparticles applied for cancer disease. But you have many, many, many other diseases with also an Upier effect, and probably with an Upier effect which is more reproducible from one patient to another than a cancer disease, which are multifactorial. And this is just one example among others. For instance, pain, when you have a pain which is associated with an inflammatory process, you can see in this publication that we published recently, and I called that yesterday, that you can design endorphin nanoparticles to target only the location of the inflammatory pain, but there are also any other example of inflammatory disease which could be treated with nanoparticles. Now, I will finish just to show you the barriers that we have to overcome to better bridge the bench to the bed. The first Barrier is the reproducibility of batch-to-batch nanomedicine preparations. The second is the complexity of the manufacturing process. I can agree with that. The third is that probably, surely, some preclinical experimental models are not at all predictive of the real clinical situation. But this is true also for any other anti-cancer compound that you have to put on the market. And in addition, there is the complexity of the regulatory barriers. And to overcome those barriers, of course, we have to collaborate with the pharmaceutical industry. But I would like to insist on the fact that it is not at all the job of the academic research to put drugs and nanomedicine on the market. This is clearly the role of the pharmaceutical industry. And in fact, as you know, the academic researchers, they have to explore new fields of research, new knowledge, to suggest new therapeutic concepts, in other words, to think out of the box. And I would like to conclude my last slide with a message to the young generation, because this is very important for the future of the nanomedicine. Last week, there was the Worldwide Congress of Chemistry, the UPAC Congress in Paris. And we had the chance to have 3 Nobel Prizes: Bernard Feringa, Jean-Pierre Sauvage, and James Stoddart. And they had a note about the molecular motors and the soft machines. But all 3, they concluded that a very important field of application of the molecular machine was the drug delivery and the nanomedicine. And there are some witnesses who attended to that Congress, and they can confirm what I am saying here. And so I am coming back to the Gartner hype cycle. I am convinced that if we are here, I am not sure we are here, but okay, probably we are at the beginning of the slope. The future of the nanomedicine is on the slope towards a maturation and a plateau of productivity. And so my message to the young generation is remain optimists, do good science, and continue to discover in the field of the nanomedicine. Thank you very much for your attention.
Thank you so much for the big progress lecture. We now proceed to the other side of the coin, how nano big can be, so to say. Thank you so much. At least it is a very inspiring lecture, and thank you, Patrick, for doing so. Together?
Selfie later.
Excuse me one moment.
How do I start? It doesn't show up on the screen. Excuse me. Hello. Hi. It doesn't, oh, here. Well, Patrick had such a convincing talk. I don't know what I can say, but let me try anyway. Let me begin with my mid-afternoon confession. Over the years, I made a lot of sins, so I want to cleanse myself before continuing. What I'm going to say today, right now, may be not accurate, not outstanding, but I'll try my best. What I'm going to say is also half semi-scientific, half unfunny jokes. Please don't take it seriously. I'm not going to be serious, but whatever you take it is your fault. Let me start with all this nano life we have nowadays. Whenever I wake up every morning, I have to start the day with my nano toothbrush, dry myself with a nano towel, use my nano lotions. We even have a nano baby. These are all you can buy at Amazon. These are true products. When you have an accident, you have to use nano first aid kit. More importantly, drink nano water. Nano water is the only one that is completely natural and environmentally friendly. Throw away your water right now and start drinking nano water. This is commercial product, so we understand. But science abused nano too. This is a book, Nanotech Pioneers. The table 2 begins with a nano age, end with a nano world. What in the nano world is nano age? I have no idea. But people use nano anyway, so I'm going to introduce myself. I am 100 nano-year-old nano man. Naturally, I have nano brain. For the last 50 years, I have a nano salary increase. So I'm a very poor man, yet I saved the money to come to Nano Valencia. Many of you know that I don't really fond of nano. The real reason is this. Whenever I order drink, they always give me nano whiskey. I had to drink thousands of them to have any effect. Something really mysterious to me is this. We have drug delivery system known as liposome, polymycel, nanoparticle, dendrimer, drug nanocrystal. We have a specific name for delivery system, but why all of a sudden we combine them and say nanomedicine? Not long ago, there was a Women's World Cup soccer. 16 teams compete in the final stage, but they're all nano nation. One nano nation won the World Cup. My question is, which nano nation won the World Cup? Answer, doesn't matter, nano nation. Same is true with the nanomedicine. Which nanomedicine was approved? Doesn't matter, nanomedicine. What is going on here? Why we abandon specific name and simply say nanomedicine? So I have to ask, which nanomedicine? Well, you know what, nanomedicine, maybe liposome. Which liposome? Which drug? Why we abandon all these specific names and call it nanomedicine? There are millions of different nanomedicine. All of them are useful? Maybe not. Why do we obscure ourselves here by just calling generic names? Not long ago, human beings believed that the earth is flat and sun rotates around the earth. This was happened before. Now, in the Homer's book, Odyssey, the Greek gods were talking to themselves. We, the gods, will live as long as the humans believe in us. The day the humans no longer believe in us, all the gods will disappear. This is even true today. Change the Greek gods to politicians. We, the politicians, will live as long as humans believe in us. Now, replace the Greek with a nano. As long as human beings believe nanomedicine, it will exist. Again, why do we call nanomedicine? Why do we specifically call pegylated liposome dox or whatever? It's a specific name is there. Why we make it very confusing? That is something I don't understand. But I have to admit that nanoscientists are very clever. The key is to translate from bench to bed. So to make this very, very fast, nanoscientists brought the bed to the bench. whatever formulation they make, they taste it right away to the patient. The results are almost always the same. Compared to control, nanoparticle functions better. Look at the result. Key is compared to control, nanomedicine is better. Regardless what nanoparticle, nano is better compared to control. Now, Warren Chan published not long ago exactly find out how much drug is actually going to tumor. One thing he did was to calculate and say, well, about 0.7%, less than 1% is going to tumor. But amount of drug that actually taken up by cancer cell is 0.0014%. Again, compared to control, it is better. But the question is whether that is enough to really improve the situation of cancer patients. This is like saying that one of the top 10 buildings in the world is Taipei 101. In a full moon, people go to the top of the building and look at the moon because they are closer to the moon than people on the bottom. 500 meters closer to the moon. That's a long distance. 500 meters. So scientifically, it is true that people at the top of the building is closer to the moon than people at the bottom. But compared to actual distance from the Earth to the moon, which is around 386,000 kilometers, this 500 meters is about 0.00014%. Compared to people at the bottom, which is about 0.00004% over sea level, look at this amazing statistics, amazing fact. People on the top of the building are 350% closer to the moon. This is scientific fact. Nobody will dispute that people at the top of the building are 350% closer to the moon than the people on the ground. Nobody argue against this because this is a fact. But in the context of the actual distance, does 350% closer to the moon, does it really matter? That's the issue. That's why all this nanomedicine paper does not tell you exactly how much is going there. Instead, what do? 100% better than control, 200% better than control. This is what's happening now. We have to look at the actual data, see whether that really makes any sense. When somebody starts saying percent increase, you have to ask for the real value. Now, what we do now is like a Don Quixote, okay? We are trying to distort the reality and fit into our own fantasy. That's Don Quixote, but science does not work that way. For example, we have a GPS system because Einstein's theory, without his theory of relativity, because the satellite moves at different speed than Earth's rotation, there's about 40 microsecond difference. If you do not account for, your GPS does not work. So let's imagine our world without Einstein. We have an immediate problem. GPS will not work. Let's imagine our world without the Beatles. They will be, I'm going to sing a song, okay? Hey Jude, don't make it nano. Take a nano and make it bigger. Well, without Beatles, well, I spent a lot of money on karaoke. So without Beatles, there will be no Yesterday, there will be no Sgt. Pepper's Lonely Heart Club Band. We have an immediate impact. Now, imagine our world without antibiotics. We really have a problem. Now, there's a bug called superbug. We don't have any antibiotic. We're going to have a problem unless we find the better antibiotics. Now, imagine a world without nanomedicine. What difference do you see? Will you see any difference? Well, individual. It's answer up to individuals. Now, the concern I have about focusing on nanomedicine and educating our young scientists with the nano, basically nowadays, whatever the research question is, answer is nano. That's what I'm concerned about. When students begin their research, well, initially they have a very big goal of, for example, understanding cancer. But as time goes on, things does not work. It just works somehow. Sometimes, some days it works, sometimes it doesn't. As your grant is expiring, you have to write and publish a paper. That's why we have lower our goal and just make a story to publish. But lowering our goal is something scientists should not do. As Lady Gaga said in her Oscar speech, what it's about is not giving up. I love Lady Gaga's, Shallow. That's a great song. I'm not going to sing it though. But it has a great lyric. Do you remember that? Is there something else you're searching for? We have a lot of something else to search for. We need to find out the real answer to all this instead of just claiming nano will solve all the problems. So basically, we should not give up. The difficulty here is that most people don't publish any data that really help us to understand, because they consider these are failed experiments, so they hide it. They just throw it away and only use the data that fits into their conclusion, nano is better. That's why I'm worried about scientists becoming Don Quixote scientists. We should not do that. Okay? We should maintain our goal of understanding cancer. We should our best to do what we are really supposed to do. I am very optimistic for the future because future will be always better than now. Otherwise, humans have some difficulty. So optimism, I'm very optimistic, but purely blindly optimistic will not help us. We have to understand what is possible, what is not. Bill Gates said, optimism requires being candid about the hard problems that still need to be solved. If there is a man who will be optimistic, that'll be him. He's saying, you can be optimistic, but you need to know how difficult problem we're facing, and we need to solve them. So blindly optimism, oh, I'm a half-glass guy, that doesn't tell you anything. You have to understand, if glass is half full, well, glass doesn't matter, but if your beer is half full, just order another one. Right? I mean, those say, oh, I'm an optimistic guy, that doesn't tell you anything. We need to understand what difficulty lies ahead, so we need to solve them. So here talking about factfulness by Hans Rosling, it's all about data. A lot of people have a misunderstanding because like a Don Quixote, every day, the same data, somebody interprets different way. But we have to look at the data itself. That's what we are talking about. Bill Gates tweet, and this is actual data. The left was data from Center for Disease Control. According to statistics, #1 killer of Americans is heart disease, #2 is cancer, the rest of it Alzheimer's disease, etc. But according to Google search, people think that cancer is the #1 killer. But according to New York Times, #1 killer is terrorism. Nobody dies in America because of terrorism. Outside America, maybe. But according to nanomedicine field, 80% of people die because of cancer. Let me ask you, by the way, why all of you do research on tumor targeting? Is that the only disease we have? Why all of you do nanoparticle for tumor? There are many other important diseases. What is the reason you focus on tumor? Because all of you believe that this is the only disease that kills Americans. There are many other important diseases. Okay? Now, we have to understand there are many other diseases we need to conquer, understand, and find a solution. So until we find a great solution, we'll have to, people continue nano, and here are my suggestions to you who continue nano research. This is the title of your next proposal. Here it is. Artificial intelligence-controlled 3D nanobioprinting of personalized human on chip with the immune checkpoint and CRISPR data mining. It has everything. No reviewer will reject your proposal. Let me tell you, preliminary data, you can use this. You can really make 3D nanoprinted human on potato chips. Human on chips is another strange term to me. If you make cells on certain chips and connect them, and someone call it human on chip, I don't see it that way at all. But I can make a human on potato chips. People, you have to think. When someone said human on chips, my first reaction was, what, human on potato chip, or what? How can anybody even call human on chips? People follow it because it came from Harvard. What's wrong with you? Think. Yeah. So until we find all the cure, it will take time. Meanwhile, until we find the real cure, let me introduce you temporary solution that can cure all your disease out there. This is not snake oil, this is real. Ladies and gentlemen, for the first time in human history, let me proudly introduce to you nanoethanol. Some people call it nano whiskey. It can avoid your weight gain. It promotes your heart health. It helps fight cancer. It improves your brain health, which I really need. It reduces risk of stroke, helps reduce stress, boosts your memory, help with digestion, lengthen your lifespan. What else do you need? Lengthen your lifespan. It is also safe with diabetes, many other diseases. I can tell you from my experience, not only nano whiskey, but also nano bourbon works too. These are real things. I drink whiskey every day. That's why you may think I'm dumb, but I'm not really that dumb. Okay? What I'm saying is, we have to change our mindset to really guide the young scientists to really expand their experience and horizon away from nano. So these are the faces of the future of drug delivery field. These are examples of faces of a young scientist who will lead the future, of course, except this guy. These are very fresh minds. Their brain is like 1 terabyte new hard drive, which can absorb anything. Why do we taint them with a nano thing? Because when they grow up as a trainer in grad school, Nano is the answer. In old days, forgive me saying this thing, okay, but we're all grown up, right? So in old days, whatever the problem you have, answer was sex. Now, whatever problem you have, answer is nano. What does it tell you? You have to do nano sex. Well, anyway, I'd like to conclude my talk using the science. The Endless Frontier is a report by the precursor of National Science Foundation. His name is Bush, to the President Franklin D. Roosevelt in 1945. This is the title of the report, Science, the Endless Frontier. Star Trek says space, the final frontier, but science is endless frontier. It's not just endless frontier. Now, we need to mention to our young scientists, explore everything that your imagination leads to. Nano is just one small, tiny field. Why do you focus on that particular space? We need to get out of functional fixedness and liberate ourselves from confined research topic. Functional fixedness means this. If you have a hammer, everything looks like a nail. If you have a nano, everything looks like a nano. Why do you confine yourself in such a small area? Somebody said there are plenty of room at the bottom. Do you know how much more room up there? I don't understand all this narrow space and claim that it can solve everything. It's not going to be that way. Next, why, as I said before, why everybody doing the same tumor targeting with nanoparticles? There is no reason to think alike and do the similar things. There are many other things to do. Why are we doing the similar thing even then? It is all about tumor targeting. We need to change this. Individual scientists does not have to follow what others do. Individual scientists must have their own voice. How? By trying their crazy ideas, making a failure, and learning from them. To avoid the failure, what is happening now is that they do the same thing that others do and have the same conclusion that nanomedicine has a great potential. Once you do that, you're safe because everybody else is doing it. But that's not how we improve ourselves for the future. Our young scientists, as I said, their creativity is unbound. Why do we make their creativity so narrow? Let them expand their horizon. So let them roam around the universe of uncertainty. The future is unknown. Future is uncertain. That's what they are challenging. The future is uncertain. Let them explore the universe of uncertainty and expand their boundary to infinity and beyond. What can be better than this? Why do we teach them only the nanoparticle? I just don't understand. What I'm saying is that we can be better, we can do better things by not limiting ourselves to nano. Universe is huge. Why do we focus on such a tiny area? Well, in the beginning, anything can be possible, but after 30 years, if your boyfriend saying, oh, we're going to get married, but 30 years later, oh, we still have a potential to get married, does that make sense? Let's move on. That's my message today. Thanks so much for your attention.
Imagine our nanoscience without Kinam. And wow, what a voice. Kinam, I'm already for 30 years, I'm a choir conductor in Amsterdam. You'll get the invitation. My favorite song with the choir, really the favorite song, it's a church choir, is I stretch my hands to you. So I will arrange it for you. I stretch my hands to Nano. Maybe. You're invited to join. Well, thank you, Kinam. And I think it's highly appreciated. Both talks are very entertaining. And now we have to see where we stand. I would like to invite you to make your vote again. Is there any change now? Please do so. I think it's stabilizing. Yes. I think no. I think the voting session is now over. We see that the differences between both opinions have increased. So I think it's really time now to start a hot debate, a nano debate, and see if that has any effect on the discussion, on the opinions. Can you go back with the slides? It's this slide. We do this with. Yeah, go back to the slides. No, to this slide. To here.
Moment that the power crash
Will not come.
no power crash.
What I propose to continue and to start a debate, 5 statements that, Kinam, are you taking that side? And Patrick is coming here. I will be in the middle. I have no opinion, or I agree with them, with both of them. Sit in the middle. And we go statement by statement. And the first statement is from Patrick. And with every statement, I first give the other debater the chance to react. And then I hope you come in. So, Patrick, can you read your own statement?
Yes. It is, I will not joke. I didn't know that it was a cabaret, you know. So my first statement is that it is not the job of the academic research to put nanomedicine on the market. If we can do, okay, but it is not the job. But to produce new knowledge, new concepts, to think out of the box.
Kinam.
I fully agree. That's my last slide is all about. Why do we confine ourselves to nano? That's what I said. I'm 100% with you. I have nothing to argue against you.
Patrick.
So we can agree.
This feels really hot.
Well, we all agree, but the disagreement is how, and the means to get there. Is it some difference? I think it is not the job of the academic research. To me, let me be blunt, because we are feeling the heat already. I think, to me, this is an excuse. We cannot simply say, oh, it's not our job to produce. Then what in the old nanomedicine about? Is it for the basic research? When nanomedicine began, specifically National Cancer Institute specifically said, we'll cure cancer using nanomedicine. That's why they spent $330 million. Now we are saying, oh, that's not our job. To me, that's an excuse, Patrick.
No, I don't at all agree with that. First of all, because I think that we have not the means at the university to be able to do really pharmaceutical development. And I think that it is really something which is extraordinary when you are looking to what happened more than 30 years ago, that a lot of people coming from the industry, academic people, are creating startup companies coming from research which is done at the bench and allowing, and I have given to you a lot of examples of startup companies who succeeded to enter into the clinic, to develop a first phase, second phase, third phase clinical trial, and even to come on the market. So you have seen the table I have showed to you. I cannot do more. Of course, I can do nano jokes.
Let them continue.
Well, if 30 years ago, when nanotechnology, nanomedicine began, if nobody promised anything, we'll just explore this, then that nobody will argue. But there was a specific goal, especially National Cancer Institute, will cure cancer. That's why most of it doing cancer research. In 1960s, President John F. Kennedy said, we'll send a man to the moon and safely bring it back to the Earth. Now you figure it out. So they figured it out. But now the goal was, we'll cure cancer, but we cannot cure cancer after 30 years. So if they did not promise anything, we know science is hard. But after 30 years promise after promise, now saying, well, this is not our job, Patrick, this is to me an excuse.
No, again, I don't agree with that. It is not an excuse. It is the reality that at the university we are not paid to put a drug on the market. Otherwise, I would enter into the industry because it is really, from a professional point of view, I think that the industry have a much stronger experience than the academic people to make pharmaceutical development. But you know, again, I would like to come back to the story with the monoclonal antibodies. I remember that after Kohler and Milstein discovery, which is a very, very big discovery, probably at that time they did also, it is not our job to put a therapeutic antibody on the market. It took a very, very long time, and I remember, because at that time I was a postdoc in Louvain, I remember that at the beginning, nobody was thinking that the therapeutic antibodies had one chance to enter into the market and to do immunotherapy. And I think that with the nanomedicine, we have almost the same story. And even with the antibodies, which is a success, you have to accept that. You don't cure all the patients. You have many, many antibodies which didn't reach really the market. But anyhow, there are very clear improvements. And I think that in the field of the nanomedicine, we are following and we should follow the same way. And this is my hope.
Well, as you said, not all antibodies treat all disease. Certain antibodies are effective to certain disease. Why do we say nanomedicine? Why don't we say certain drug delivery vehicle? Nobody said we have an antibody that cure everything. There is a specific antibody.
Yes.
Nowadays, antibodies turn into nanomedicine. You just attach drug, it's nanomedicine, right? By the way, do you know why they put him over there, far away, so I don't beat him physically? He doesn't beat me. This is why we're so far away. But I think it's not about, I don't disagree with at all. Just simply, nanomedicine area promise way too much than they can deliver. And now it's saying science is hard. From the very beginning, they should have said, science is hard, we'll try, we'll explore this area. If it doesn't work, we may just change the direction, find a better way. Now we keep going the same direction. After 30 years, none of the nanoparticle clinical study worked. There, as Patrick said, preclinical model may not be representing what's happening in humans. Then we need to find a way to find a better clinical model or better understand why you keep continuing nanoparticle tumor targeting still. Somebody can answer to me. I don't understand.
Well, maybe somebody in the audience would like to participate for a couple of minutes, or a few of you. Is somebody there who would like to say something? I hope there is a microphone. Yes, there is.
We have a better light for picture now.
Yeah. Microphone don't work.
I think maybe you can hear me. And it's a very entertaining session. And there's a number of critical issues are missing. And for instance, the one thing is the theory talked about EPR, that all the tumor has a blood flow similar to normal tissues. But in essence, there's a great magnitude of vascular embolization, thrombogenic embolization. And the last June issue of Blood showed an excellent review, actually the original work, and 37 million people, the work was retrospectively analyzed, and the thrombogenic potential is about 6-fold higher in cancer patients, 1 month before the diagnosis, indicating blood flow is very much blocked in the tumor tissue. So there's no EPR without blood flow. So that's finished. The another issue is about
But we have to focus on the statement.
yeah, and then
We end up everywhere.
but this is about the antibody, for instance. It works in the monoclonal model of a small mouse tumor, which nobody works more than 2 centimeter tumor because of the sort of the regulation. But the targeting to the larger tumor has so many, or the natural tumor. You cannot work in the model of particular syngeneic model because there's no immunological heterogeneity. Human tumor, for instance, the kidney tumor or lung cancer has more than 500 or 300 mutant in the same patient, which, the Bart Fogelstein in Baltimore has shown. So many mutants in the single patient, 100 mutants. So how do you expect 100 mutants can be covered by a single monoclonal antibody?
Yeah, it's a very important remark, and biology and tumor biology is very complex, and how to utilize this for medicine, but I need to stick to the statements, because this statement is not about scientific contents or biology so much, it's more about the task of academic research.
Because of the academic research or everything is so much under the regulation of the authorities. You have to go for ask the permission of the Pope, and Pope may say, okay, and then this you can do it. But unless you get the approval of the Cardinal or Pope or whoever the authorities, you cannot have practice in the small scale individual your ingenuity. You have to, everything has to go through the approval, and the academic has not much enough money and power, and the scale is so small, so nobody believes a few cases unless you run hundreds patients or thousands patients, and
maybe
that's
that
not
relates
possible.
to that. So the clinical translation is costly and complex.
Yeah, it's so complex.
And that is related indeed. Thank you for this
Exactly.
statement. I have to run for the time, otherwise there are 3 and not more, but please, can you be short? Also you, Gezi?
I never can be short. Okay. First of all, I think it was fascinating to hear you two. Everyone give a great lecture. I like to get them so I can discuss them with my students. They are terrific. But I think it's one very good example in a dispute that both sides are right. We have to admit, and I think the problem is that each side go too much to the extreme side. But basically what we have, first of all, academic can never develop drug.
Of course.
Really does not nano on anything. Simply, it's not the rule of the academy. Academy has to come with the idea, with feasibility, then it go to startup, to big company, and they develop it because they are much more professional. It's a wasting money if academy will do it. But I think so that's really obvious. But the point, Kinam, is that you really throw the baby with the water together, in a way, because nano has a limited role, and we should all agree about it, no question. But this role can be quite really important in quite many cases. You know, I'm one of the few that developed a nano drug, and you are aware of it. But I want to go to Ambisome. Okay, Ambisome, which I was not involved in at all, it's a fascinating drug. It really saves so many lives, and if it would not be nano, it would not
just a second.
be working.
Now, if you're saying Doxil and Ambisome are nanomedicine, I cannot argue against it. But
Okay.
that was developed a long time ago, right? I mean, do you call it nanomedicine? Doxil and Ambisome, they were developed several decades ago. Now you're calling it nanomedicine? That represent nanomedicine? Because there are 2 products, all nanomedicines are okay? I mean, that's the argument I don't understand.
No, for me,
No, I don't agree with that.
We go into semantics, and semantics is,
Yeah.
we called it liposome at that time, and they are small liposomes, so they are under the umbrella of the nano.
Of course.
You can accept it or not, but that's fact of life. They are defined. Okay, I agree with you that it's better to call the man by its name, to call him John, and even the family name, John Silver, and we know who is are. I agree with you fully, and that's why I said that both are right. But on the other hand, nano has a rule and can do things that other drugs cannot do. And actually, if we look at the success rate, you know, the success rate, you said 1 in 10,000, but once a molecule get into animal, the success is 1 in 1,000. One is going to animals. And in the nano, the rate is 2.5 per mil, 2.5 times better than the regular drugs. So I think that that's really a fact that we all have to accept. That's really fact of life. So
Okay,
I think
thank you. Jesse,
Yes,
thank
but
you
may
because
I-
real time. You want to
Yes, I
give
would
a
say
nano
that
answer?
I don't at all agree with Kinam saying that liposomes are not nanomedicine. Then what is your definition of nanomedicine? Because liposomes have a bilayer of phospholipids and lipid nanoparticles, they have a monolayer or what else? How can you say that you have a difference between those all different types of nanotechnologies? For me, and this is the reason why I started my presentation with a clear definition of what is nanomedicine, and this is a definition which is accepted in Europe, is that when you have nano-sized particles able to modify either the bioavailability, the pharmacokinetic, the biodistribution, to improve the therapeutic index or to reduce the toxicity, this is a nanomedicine. I didn't invent the name of nanomedicine myself.
I'm asking, which nanoparticle? Why don't you be more specific? Which nanoparticle affect the PK profile? Tell me exactly which one, then I will accept it. Why do you keep saying nanoparticle? It's just like saying human beings are here. You are not a human being, I am, but I have name, you have name. Why don't you call our names? Why don't you say, oh, human being is standing there, another human being standing here? What's the point? Why don't you have a specific name?
This discussion is now about underlying terminology and nanoparticle and whatever. This is not my intention.
We are rolling on the words.
Sorry for you, otherwise we will never make it. I would like to go to statement 2. Maybe you can react to that. Let's go to, can you push the button, Patrick?
What?
It's not your task, it's my task, but can you push the button on your, for the next slide?
Kinam.
Yeah, it's a statement of Kinam. Kinam, can you read the statement?
The whole world has been obsessed with a nano. A change is needed to make this field more creative. I mean, that's what I said. Again.
sorry, Kinam. The word is now to Patrick because you gave your lecture. We know this.
Yes, I don't agree with the first part of the sentence, because personally I can tell you that I am not an obsessed guy. I am not obsessed. I am not obsessed by nanosex. I am not obsessed. I am not obsessed by nanoethanol. And so I can tell you that the first part of the sentence, I don't feel that it is my problem. The second part of the sentence, the second part of the sentence, okay, I agree, of course. We have to, and this was a conclusion of my talk, we have to make the field more creative. And this is the reason why the PhD student, the postdoc, the academic people, they have to find new way in the field of the nanomedicine, more creative, thinking out of the box, etc., etc. And I agree that it is very, very important. And I am convinced, and I have given some example about that, that there is a room, plenty room for the young people, for the next generation, to find new way to still improve the activity of the nanomedicine. So I can agree with the second part of your sentence.
I thought compared to American, French people are obsessed with wine, obsessed with soccer, obsessed with the dress, clothes, obsessed with everything. So when French person said, I'm not obsessed, I don't know how to take it, really.
Well, lucky enough, I'm Dutch. Let's go on to the next question. Please.
Hello? Oh, hi. So I do agree that there needs to be a change in this field. 2 changes that I think there should be is 1, we need to have proper controls. In the clinic we control against standard of care, but with animals, for some reason, we don't do that. So how is it that we can translate our technology and say that our technology is groundbreaking and translational if we don't implement the proper controls? But secondly, I think it's a problem with the system. I mean, I'm a young scientist, but then I'm not given merit to make mistakes. I'm not given merit for my negative data, right? Whereas we can all agree that negative data is incredibly important. So I'm not given merit to be creative because this is no longer the age of Madame Curie. This is no longer the age of Tesla. This is the age where we apply for grants and productivity is actually determined by our politicians.
The winner of the debate. Any response to this?
Well, I'm really impressed for your comment because that's exactly what we need. Right now, to you and everybody else, change seems to be very difficult, impossible. But look at Nelson Mandela. One person changed the whole South African political system. Until it is done, everything seems impossible. But a person like you, a good starting point to start changing.
Well, thank you. I think I go to the next statement. We have 5. Can you push the button again?
Okay. Here is my statement, yes. So now it's close to the preceding one. Nanomedicine research should be improved to overcome the current hurdles.
We actually discussed this quite a lot, eh? Maybe you have a response, Kinam?
Obviously, it has improved because nothing much has been done. All they have is potential. You see, the problem with the potential is this. I have a potential to become a billionaire by winning the lottery. You have the same potential. But we don't live with the potential. I don't give up my job because of the potential that I may win the lottery. Potential is potential. We need to translate into reality. That's what's missing. To really improve, overcome the hurdle, my message today has been, why do you focus on your nano? That's my question. Why do you focus on your nano to overcome the hurdles to solve all the problems?
Yes, but my answer is, why do you focus only on anti-cancer compounds? Why are you focusing only on hydrogels? Why are you focusing only on anti-cancer, on monoclonal antibodies? Of course, you have always to focus on the clear subjects.
I'm sorry, only focus on what?
Clear subject.
On a clear subject.
But it's not clear to you, that's clear.
When you are starting with a PhD, again, I am speaking for young people, you are starting with a project and with a focused subject. If your subject is in nanomedicine, well, you will continue in the field of the nanomedicine until the defense of the thesis. If the subject is about monoclonal antibodies to improve the immunity of the melanoma, well, you will continue with monoclonal antibody. If the subject is hydrogel to have a slow delivery of this type of peptides or otherwise, you will continue with that. I don't understand what you mean by focusing on nano.
Oh, I'm not accusing anybody. I'm simply saying if your research topic is nano, please go ahead. My question was, how come everybody in nanomedicine field work for tumor targeting when there are many other important diseases? Why? Why do you focus on specific topic? If we 10,000 people doing 10,000 different research topics, some of them may bring real breakthroughs that'll help us all, like Einstein's theory of relativity. Why everybody focus on tumor targeting? That's my question.
So maybe a reply from the audience. Can you react?
First of all, Justin Haynes, Johns Hopkins. Kinam, you'll notice that I'm on Patrick's side over here. But I promise I'll buy you thousands of mini nano whiskeys to make up for this. But first of all, I feel like I have to call you out on some alternative facts that I feel like you're asserting. So to say that everybody does it, you said 4 out of 5, that's probably a result of the cancer field putting as much money in the US as they did into the research, then you're gonna have papers follow. So I think that's a little bit natural for people that have the money, that's where they focus their work. But I think the nano things that they do there can help in other diseases. So. But secondly, I think that in a way, you said that if you count Doxil and other polymeric micelles and so forth as nanomedicine, then you can't win that debate then, right? So I think in a way, you've given up that if nano does include those things, then Patrick should win. So having said that, and you say that we shouldn't call a liposome nano because it's a liposome, we should be very specific. And you listed about 10 things or so that we're now calling nanomedicine. If you think about how many different steroids are there? How many different NSAIDs are there? How many different small molecule chemotherapeutics are there? Do we call them all by their name when we want to refer to them, or do we say a chemotherapeutic or a steroid? Or let's put them all together, we call them small molecule therapeutics. What about macromolecule therapeutics? Do we call them all by their individual, peptide name or protein name or nucleic acid name? We call them sometimes macromolecules. Now, is it useful to call them individual names? Maybe. But if you wanted to separate this room, women on one side, men on the other, would you then say that you should say every woman's name to go to that side, one at a time? Or do you say all women please go to that side and all men to this side? So you would use a generalization. What all those things that you listed have in common are they all have a size in common, just like small molecules, just like macromolecules. They all have nano in common, so it makes sense to call them nano. So in fact, you made the case, since the 60s and 70s, we've had products. We've made enormous progress. And then the question is, do we have, promise into the future? And it's come out that 2.5 times better rate of getting a product onto the market if you use nano than if you do not. So I have to say that I feel like if that's the case, then maybe you conceded.
Thank you. Shall we leave it? Because I have got an extension because the time is over, but we will finish. We have 20 minutes more.
I cannot say anything against Justin, so I'll let
Good. Patrick, your turn.
Next one.
Yes, you learn quickly.
May I answer?
You have to read the statement.
My comment is nanomedicine scientists behave like politicians.
Do you think I am looking like a politician?
Yes.
So the discussion will become very, very difficult.
I think my point is, politicians, look at politicians. They promise everything. Our lower taxes, if we have a Brexit, you will have more money in your pocket. They promise everything. When things doesn't happen, look what they do. Look, the other party blocked me doing so. They always blame other people, right? If nanoscientists, well, I should not generalize, if many scientists promise something and doesn't deliver and blame some others to make an excuse, what difference do we scientists have from politicians? We should not promise anything. We just promise that we'll do our best. We cannot promise that we'll cure cancer. We just do our best. But nanomedicine, again, the whole idea is to curing cancer. That's why National Cancer Institute spends so much money over the years. Do you think the National Cancer Institute has so much money, they just want to give you money for your fun? No. See, we need to be a scientist. Scientists should act like a scientist. Do not promise casually. You cannot cure cancer now. No matter what you do now, curing cancer will be very difficult. So we cannot promise, we just promise, I will do my best. But if politicians say, I will do my best to improve your life, he will never get elected. That's why they just overpromise all the time.
But what you are saying is true for any domain of the science. Because, you know, in my opinion, the scientists, the true scientists first, they have to dream. When I was a young PhD student, I was dreaming about the possibility to use nanoparticles to deliver drugs or compounds intracellularly. So when you are dreaming, you are convinced about what you are doing. And if you are convinced about what you are doing, of course, you do some promise. But in general, I will try to do this, to reach this therapeutic goal. So if you have not that in your heart, I think it's very difficult to advance.
I don't think you have to separate having a dream. That's all humans have. That's why human beings become dominant species in the world. Humans have a dream. They conquer their dream by doing something. But the problem I have is that you promise before you have a dream. Dream is good. We need to have all these dreams, conquering everything. But then promising before you do anything, that's what I'm saying here.
Well, two people, two scientists, replying to what you have said about dreams and promises.
I'll just speak very loudly. I'm tired, and I think we need to reach a compromise, and I think in a sense of...
No. Yes. So I think the problem is we have to sell our projects to the funding bodies, and I think that's mainly a voice that we're missing maybe here today on this debate. We have to sell our ideas and our projects to the funding bodies in a sexy manner. So using all these words that Kinam put together in this final title, that helps get the projects funded. But probably that's promising way too much than we can actually accomplish. So how do we reach that compromise? And another point I wanted to add to this is last year I remember we had a session with Professor Jain, and he told us young scientists the research world is developing super sexy, fancy, fashionable new drug delivery systems that no one will be able to pay for. Not the patients, not the national healthcare systems, no one. So where do we reach the compromise here?
Patrick?
No, I can agree with that. That's true that the scientists actually, they are depending from the agency who are releasing the money. So of course that we have to present a project in the best way, in the more intelligent way, in the more optimist way. But this is, you are doing the same, Kinam, I am sure. Or otherwise you have no money in your pocket to pay the PhD students and the postdocs.
I'm not French. Well, I think that's a good
You are a
point.
funny guy.
When we talk about compromise, just like a politician need to compromise, scientists need to compromise too. Here, we intentionally show the extreme view, but it's you compromise yourself. Just choose what's best for you, and you just continue with your research plan and your vision. We just present you two extreme view, and compromise becomes a dirty word in political world. But we really need to compromise. Compromise based on your pathway, your future. You need to compromise what is best for you. Individuals are different. We cannot say that this is the best way for everybody. Everybody is different. That's why we're saying it's up to you. Okay, we just present our views. Who knows, he's 100% right. But that's okay. Always we need to have an opposing view. That's where we think, where we progress.
Well, Professor Pepas.
Kinam, Patrick, you have been good friends for 35 years. This has been a magnificent presentation. It made me think. I hope it made other people think. But I want to talk to you not as a politician, not as a professor, not as a scientist, but as a patient. What you're doing here today is a very nice exercise from which I have learned a lot. But as a patient, I want an answer now. My best buddy at Purdue University, where you are, passed away last week for pancreatic cancer. After 5 years of having a big hole, a macro hole, in his body through which all kinds of chemicals were thrown. Arvid Varma is dead. And there are many other people like him. And we are sitting here and saying, you know, NCI, you're right. Turn off this particular door. Don't give any more money for nanotechnology. You know why? Because Patrick Couvreur didn't come up with a new drug. And I disagree with the gentleman who said earlier, academia should not be doing drugs or is not doing drugs. Have you ever heard of a man about 70 years old in Cambridge, Massachusetts, who has come up with about 1,000 different patents? Please don't tell me the universities don't do real work. I speak from the patients. We cannot just stop the discussion. This is not a Byzantine period discussion about whether Virgin Mary was virgin or not. It's time to solve real problems. And if the little we understand about nanomedicine has helped in the case of doxorubins in Kinam, we should admit that that's nanomedicine. I don't care if it was the last 20 years or 50 years. It's nanomedicine. When Chad Mirkin or somebody else comes up with something, the SNA, that will pass through the blood-brain barrier, I applaud him and I want to give him more money. Thank you.
Thank you for more emotional comments.
Just a couple of comments. One is that, Kinam, you're a good friend of mine, but I feel like you've been using straw man arguments in many cases here. Number one, saying that everybody's into nano. And if you look around this room, I don't even know if that's true. I certainly know in my own department that we have 1 or 2 people who are doing nano delivery out of 8 or 9. Now, it's not. I just don't see it as being all that dominant. But having said that, I'd also like to point out that there's a lot to nanomedicine that is beyond just imagining that you are going to have a particular concept of for nanomedicine, and that's going to cure something. It rarely works that way, even with regular drugs, small molecule drugs. Usually people come up with some general idea, something works, and often it's not the thing that you started off with that actually works, it's something else that comes out of it. And so, in a way, I feel like we don't want to just sort of cut off a general area of research because it's been unproductive, because it probably will be productive in ways that we can't even predict at this point.
Thank you. Well, I have to come to a final debate moment, and actually there's one slide left which I have been putting forward, and actually I don't want to. Can you?
The next one.
Nanomedicine is indeed oversold, but nonetheless not less successful. I would like to not to have a debate on this statement anymore. I think it's rather a conclusion from the moderator. There are products on the market, liposome products, monoclonal drug conjugates. There are viral vectors for gene therapy. Doing maybe not what we want them to do, but they are helping, maybe to a very limited extent, patients. And we want much more. We want to dream, we want to promise, and we want to deliver much more. I think science is a step-by-step process, and as human beings, and also as scientists, we should remain very modest. So I would like to end this session with these words, but first we have to vote. Please vote. Please vote.
About the sentence?
No,
about the status, big progress versus nano-progress in the field of nanotechnology, nanomedicine, we should say. We cannot vote? I will go to the responsible.
The vote is not working.
People.
It's working?
There is a problem. Can you help with the problem? Or are you leaving? You help with the problem. Okay, because this whole voting is out of my scope. But maybe we can only vote one time or something like this. Well, there is some confusion about the voting system. We cannot do this. But I have a more clever way to do it. I have a dream, always a dream. I had a dream to shake hands with both of these guys. When I'm doing that in the middle of the floor, then I will ask you, big, stand up and wave. And then I ask you, nano, stand up and wave. And then I will judge how much 40% is, or whatever. Please stand. I will guess. How much nano? 30%? Nano% he's saying. No, I think about 30%. 30. Please sit down. Some people never vote. They have no opinion, so remain seated. But who is voting for Patrick? Big progress? Oh. That's the other 70%. I think the nano debate mattered a lot. And I hope you enjoyed this. I did. Maybe next time in Las Vegas. Thank you. Have a good evening.
Thank you, Mr. President.